Hi, this is Ulysses Balis from the Department of Pathology, Division of Pathology Informatics at the University of Michigan. Joining me is Dr. Jason Hipp, who is the Informatics and Image Processing Fellow here in our division. In the next few moments, both Jason and I are going to share with you the uses of spatially invariant vector quantization on a large macro scale, in this case remote sensing, and Jason is going to describe the image under investigation.
This is a satellite image of the NIH campus using Bing Maps. You can see here in the dot is the center of NIH campus with building 10 right here. To the left here is Suburban, and to the right over here is National Naval Medical Center, and south is Bethesda. And we're going to generate a vector.
Actually, several vectors. Again, the advantage of SIVQ over other contemporary specific algorithms is that it is domain independent and can be programmed with relatively little, essentially no, in fact, programming expertise. Only what is required is understanding of the subject matter. In this case, we're looking at a remote sensing image. I'm going to generate a simple vector of various degrees of healthy and unhealthy lawn. In this case, this is a moderately healthy lawn. I'm going to generate a single ring vector, and by selecting this ring vector, this is the circular vector that is generated, and because of its circular nature, what would otherwise be 700 to 800 million degrees of freedom can be collapsed to a single search predicate, taking advantage of the continuous or perfect symmetry that the ring structure offers in 2 dimensions. With that single circular search predicate vector, I'm initiating a scan, at which point now the image is iteratively being queried with all the possible orthogonal permutations of that single vector, generating a heat map. As you can see here, we're having some lawn which is matching this particular healthy predicate, and the heat map, of course, with red corresponding to high level of statistical match, and yellow less, and then green even and less. This then allowing for a very quick quantitative assessment without any specific programming to features within the image that match the original search predicate. This algorithm similarly can be scaled to multiple processors for enhanced speed. This is actually being carried out on my Sony laptop, giving an idea that computational power is not a significant requirement. Here we have, for example, in the area original to the search predicate, a high level of match, showing again a way to very quickly generate a foreground feature selector for healthy grass. I will re-carry out this search with a separate predicate, the next query being not so healthy grass, and we found, for example, earlier on some areas of parched grass that I think would be a good fit, perhaps, for example, right here being a good predicate. So we'll search for that, and now we'll scan the entire image for that not so healthy area of grass. And ideally, in short order, we should have areas only corresponding to the less healthy grass and not the healthy grass. And in fact, we can see that we've avoided the healthier grass and are now selecting for ideally unhealthy grass. So this area should come up, if our algorithm is working correctly, relatively high in the heat map scale. Again, we found some unhealthy areas here, some unhealthy areas here, and here. So the algorithm seems to be working, again, avoiding all what we previously called healthy. And our heat map, in fact, is finding the unhealthiest regions. So that's good. So in summary, this is an algorithm that does not require significant image processing, computational, or advanced mathematical subject matter expertise. Rather, it requires subject matter expertise of the images themselves which are being reviewed. And Jason will go over very quickly the high points that set this algorithm aside from other contemporary specific use case algorithms.
This SIVQ is a standalone program that is capable of running on a laptop. The advantages are that there's no training required, and it can work with any image format. Images downloaded from the internet, digital pathology slide images. It's easy to use, and brief tutorials of approximately 10 minutes can get a person up and running in using SIVQ. As you can see, it's very specific in identifying features, and it can work at different length scales, allowing you to zoom in for higher magnification of particular features and lower power assessment. And the other advantage is, as you saw here, we did 2 vectors. We can add and subtract different vectors using Boolean logic.